Hi everyone, welcome to this M2D2 talk. Our speaker today is Paula Marin Zapata, and she'll be talking about her recent work about how to use cell morphology to guide de novo redesign in drug discovery. A bit about Paula before we start. Paula is a data scientist at BioAG in Berlin. She obtained her PhD in biology from the German Cancer Research Center DKFZ. In 2017, she joined Bayer as a postdoctoral fellow, where she developed a deep learning method for phenotypic profiling in plant sciences and cellular imaging. Since 2019, she's working in the machine learning group at Bayer R&D, where she focused on image analysis application to drug discovery. Thank you so much, Paula, for accepting to present your work here today, and looking forward to the talk.
Thank you a lot, Prudencio, for the invitation. And I'm very excited to share the work with you guys. And just so that the talk is more interactive, please raise your hand and ask questions during the talk. I will be happy to answer questions and be interrupted. So welcome, everybody. Today I will be showing you our work where we use cell morphology to guide the de novo hit design. And so starting with a general introduction, so in the field of computational molecular design, we propose new molecules either from de novo or so from scratch in de novo design or from pre-generated libraries such as in virtual screening. And we use computational methods as scoring functions to help optimizing and predict properties of these molecules. And the properties that we could predict can be structural features, physicochemical properties, compound quality, and so on. But we could also predict activity against biological targets. However, the problem or like the main difficulty about predicting activity against biological targets is that it is very difficult to incorporate biological data. And there are many reasons for this. The first one is that sometimes we don't even know which biological readout is relevant for the problem that we have at hand. If we want to merge data from different sources, we know that biological data is highly heterogeneous and is very difficult to compare. Or usually there are insufficient quantities for machine learning models. And finally, of course, biological systems are very complex and they span from the full individual level or organism level down to the cellular and subcellular level. However, recently there have been some technologies that at least tackle these problems at the cellular level, and these technologies are called cellular profiling technologies. And what these technologies do is that they allow a systematic and large-scale characterization of the cellular response to perturbations because they take a holistic view and they measure hundreds of parameters at the same time. So the idea is that by using these technologies, we treat the cells with perturbations. It could be chemical perturbations or genetic perturbations. And then we use omics technologies as readout to characterize the effects in the cells. And you probably know many of those technologies, such as transcriptomics, proteomics, metabolomics, and so on. But let's say that the new kid in the block is called morphological profiling, where we simply take images of the cells and then assume that the effect of the perturbations inside the cells can be encoded in the morphological changes that these perturbations induce in the cell. And of course, these type of technologies offer a different let's say, interpretation level than transcriptomics, because we cannot directly relate the changes to a gene, but it offers many advantages of the other technologies because this is among all the most cost-effective and high-throughput technology, and it's very accessible because most labs and companies already have automated fluorescent microscopes, which is the only thing that you need to do to perform this assay. And just so that you get an idea of what throughput you can get with morphological profiling, at Bayer we can sample around 9,000 samples per week using only one microscope. So it's really, really high throughput. And so how morphological profiling is usually done is done with an assay called cell painting, where we literally paint cells with fluorophores of different wavelengths and then image different constituents of the cell with fluorescent microscopy. And in this assay, we use 6 stains to image in 5 fluorescent channels to stain 4 organelles inside the cell. So in the first channel, we look at the nucleus, the second one, the ER, then mitochondria, actyl, Golgi, plasma membrane, and nucleoli and cytoplasmic RNA. And how we use cell painting data usually is that after we obtain the images, so here on the top, you see a zoomed in version of the images of the cells. And in the bottom, you see a zoomed out version where you contain many cells in one image. And how we usually use this data is that we first pass these images through a feature extraction pipeline. This is currently done with Cell Profiler, which is a traditional or like a handcrafted feature. It's a feature extraction method that consists of handcrafted features, but these features are extremely good. And therefore, we usually just use directly the feature vectors representing these images instead of directly the images. And these feature vectors are known as morphological profiles, and they, as I said, constitute the basis for all further analysis of this type of data.
One quick question, Paula. If you go back to the previous slide, I was wondering if there is anything else you can measure using this technique, like a particular type of protein, or it's just like cells component that you can measure using this technique. And I'm just a newbie in the domain, so that's what I'm asking.
You can measure whatever you want with immunofluorescence. So if you have an antibody against your specific protein and then a fluorophore against that antibody, you could virtually image any constituent, like, or any specific protein. But the idea of this assay is that it's fixed what we looked, because we want the data to be reusable. So if for each experiment or each project you change the set of stains, then the data is not comparable anymore.
Okay, I see. Okay, cool. Perfect. Thank you.
Yeah, and the idea how you use this data is that you will have a database of morphological profiles for which you know what they are doing inside the cell. So, for example, you have profiles of toxic compounds or profiles of mTOR inhibitors, and at the end, guilt by association, you can generate hypotheses of what new perturbations are doing inside the cell. So if the profiles are very similar to a group of toxic compounds, you can say, okay, maybe my compound is being toxic.
Okay, perfect. Makes sense. Thank you.
All right.
Can you give us an example of how the vector of features look like?
These are real features, one feature per cell, and then we take the average. But the features are, so first of all, we segment single cells and we segment 3 components for each cell: the nucleus, the cytoplasm, and the full cellular area. And then for each compartment, we extract hundreds of features. So for example, intensities, texture, heraldic features, colocalization between the channels. We also extract channel-independent features such as shape and size. Things like that. Is that what you mean?
Yes, thanks.
All right, and then what we asked is, okay, so we have these big data sets generated with cell painting. So the question is, how can we use profiling technologies to guide the design of small molecules with a desired biological activity? And looking at some inspiration, so most of you might be familiar with this, with the recent, but not so recent anymore, generative models, where you, for example, input a text, and then the generative model produces images. And these are examples of DALI or diffusion models, where if you put as input text an armchair in the shape of an avocado, you get these synthesized images, or below for an oil painting of a space shuttle, you get these images. So we thought, well, what about if we use a similar approach, or at least a similar reasoning, but instead of feeding text, we feed in morphological profiles of cell painting images, and instead of asking for images, we ask the model to produce chemical structures. And in this sense, we will be tackling directly the de novo molecular designed field, proposing novel chemical structures that satisfy the input desired morphological profile. So as an example, if we put the profile of an mTOR inhibitor, then we will expect the model to produce molecules like taurine-1 or rapamycin. And to do this, so to tackle this, we decided to train a generative adversarial neural network conditional morphological profiles. And this is the overview of the model. So we have a generator network that takes as input Gaussian noise and a morphological profile and produces a molecular embedding that represents molecules, and I will talk about this embedding later. And this generator is trained in an adversarial manner with a discriminator that discriminates if this molecular embedding is from a real or from a generated molecule, and therefore forces the generator to generate real-looking molecules. And it is also trained to maximize the prediction of a condition network, which takes the molecular embedding together with the morphological profile and predicts if they match each other or not. And how the molecular embedding is computed. So this was done with a variational autoencoder to reconstruct selfie representations of molecules. And it was based on recurrent neural networks, and it was trained in Campbell 21 using 1.2 million molecules. And this embedding was trained. So before we even start training the GAN, and at the end, once this model is trained, we just use the decoder to decode from the molecular embedding into selfie representations, and then interpretable molecules. And then the other part of the model is trained together. And to train this part of the model, we use a public cell painting dataset that is called the BBB-SEED36 from the Broad Bioimage benchmark collection. And this dataset contains 30,000 compounds with their corresponding morphological profile. Yes, I see a hand.
Yes, one quick question. How do you deal with the error propagation in the autoencoder? Because like the autoencoder is not digestive, right? So like you can end up with an embedding and there's no molecule that corresponds to it, but at least and vice versa. So how do you deal with the small error that the GAN make, and make sure that it decode to something that's reasonable and not, and that's because it can decode a slide that's invalid or anything as well. How do you deal with that?
Yes, we didn't deal with that. And you will see that also the validity of the model is not extremely high, but still the representations are very good. And at the end, we were focusing more than in having a very good generative model, in really proving that the morphological conditioning is doing something. But yeah, that's all right. So and then in the rest of the talk, I will show you results for the evaluation of the model. So first, the evaluation of the overall generative performance. Then show you how we can show that there is a translation between phenotypic similarity to generated chemical similarity, and we show how we provide evidence for the generation of bioactives using profiles from genetic perturbations. And finally, if there is time, I will also show interpolation experiments in both the phenotypic space and the chemical space. So starting with the overall generative performance, we condition the generator on 30,000 randomly selected profiles per condition group, so from compounds and DMSOs, which are negative controls, and computed several properties of the generated molecules. But I don't want to go too much into detail into this table, but what we can see is that we have an okay validity, and this is what I meant prudential. So we only get a validity of about 50% of the molecules. But in compensation, we have a very high uniqueness of the molecules, so 99% uniqueness. So we also see that the molecules are novel and diverse, and that even if we also didn't account for synthesizability while training the model, we can see that 50 to 70% are synthesizable, judging from two synthetic accessibility scores. And at the end, the main conclusion from this table is that the model is able to generate drug-like molecules with drug-like characteristics with some reasonable performance. But at the end, this is not, as I said, this is not the interesting part of the work. The interesting part is to see if there were really, if this conditioning is doing something to the chemical space or not. So at the end, during inference time, once we train the model, we forget about the condition and discriminator networks, and we only work with the generator. And and we feed the generator with different morphological profiles from different compounds. And we ask the generator to generate an arbitrary number of molecules for each input condition. So in most experiments, we're usually generating thousands of molecules, but here I only show two because it's not possible to show more. Interesting question is, are there any sensible relationships between the input profiles that we use to condition the network and the output chemical space that comes from the model? And to answer this question, we decided to go for a clustering-based approach, where we initially clustered the morphological space using 20 clusters. And here on the left, I'm showing a minimum spanning tree, which basically recapitulates the hierarchical organization of those clusters in the high-dimensional morphological space. So if two clusters are adjacent to each other, this shows that they are also closer in this high-dimensional space. And what is important to know also from this figure is that we selected cluster 0 as the cluster for DMSO controls, so cells that were not treated with any control, so the healthy or untouched phenotype. And then we numbered all the other clusters by increasing distance to this inactive cluster. And that means that the higher the number, then the more distant from the inactive DMSO phenotype the cluster is. And yes, and then what we did was to pick profiles for each of these clusters and generate 30,000 molecules per cluster. And in order to find relationships between the two spaces, we took cluster 0, so the inactive phenotype, as a reference, and then started to see how the distribution of similarities in the chemical space changed as we moved away from cluster 0, away from this cluster in the phenotypic space. So maybe with this figure, you can see it clearly. So on the x-axis, we have the Morgan similarity of generated molecules relative to molecules from cluster 0. And then when we start moving away from cluster 0 along this line, which is this line in the phenotypic space, we see that the higher we go with numbers, so the more distant we go from DMSO profiles, the distribution of similarities with respect to cluster 0 move toward lower values. So the chemical space is more divergent, as we also diverge in phenotypes. And this is not only true for that branch of the tree. If we go through the second branch, we see a very similar behavior. And if we go to the third branch of the tree, we also see that for most clusters, these distance relationships are consistent. And as another visualization of basically the same results, we also took nearby clusters and then from the minimum spanning tree, and projected the generated chemical space using TMAP projections. And what we see is that if we take consecutive or adjacent clusters in the phenotypic space, the chemical space is not very well differentiated. However, when we take distant clusters in the morphological space, then we see that there is a clear segregation between the two groups in the chemical space, which is also very encouraging. So at the end, these results show that the model is able to translate morphological similarity into chemical similarity, and that it even presents distance relationships between both levels. All right, but at the end, the interesting question is, can this model generate known bioactives for specific targets or not? And before I continue with this part of the results, I want to highlight that during training, we only use pairs of morphological profiles and chemical structures, but at no time we gave information to the model about which targets these compounds are targeting inside the cell. So what we thought to answer this question about generation of active molecules, we thought, so okay, what we can do is that instead of feeding profiles from compounds, there are also public data sets of genetic perturbations. So we could feed a profile from a genetic perturbation for gene X, like let's say, and as the generator to generate molecules conditioned on that profile, and then search if among the generated molecules we can find known agonists or non-actives against the input gene. However, this is a very dangerous approach to do anything, because as I told you, you can generate as many molecules as you want for each input profile. And of course, the more molecules we generate, the higher the probability that we'll find an active among those generated molecules. So to try, like, we really try to do a fair comparison. So what we decided to do is to first do, to generate molecules, but then first do a scaffold enrichment analysis where we search for overrepresented scaffolds when conditioning on the specific gene compared to DMSO. And then only check if among those enriched scaffolds, we could find non-actives against the gene. And for this part of the study, we used as input profiles, we use genetic perturbations from the so-called BBBC 37 dataset, which contains like genetic perturbation, so gene overexpression perturbations for 220 genes. And to search for actives, we search in the ESCAPE database. But in this database, we only searched for agonist compounds, following the idea that overexpression perturbations should follow agonism and not antagonism. And this was kind of unfortunate because since we were restricted to agonists, we could only fetch reference compounds or known actives for a set of 10 genes out of the 220 genes. And not all those genes were amazingly reproducible in the phenotypic space. So here in the bottom row, you see the projection of the phenotypic profiles for these genes. And you see that some of them are not very distinguishable from the controls. So in gray and in black, you see the controls from the training set and from the overexpression set. For example, for TP53, you see that they are not very distinct, like separate, they don't separate that well from the controls. But for example, for BRAC1 and for NF-kappa beta 1, then we get a, or from HSPA5, we also get a decent separation. All right, so as I said before, so the approach was, we have these genetic perturbations, we generate molecules, perform a scaffold enrichment analysis, and then check if those enriched scaffolds are in any of these active compounds against the genes. And these are the enriched scaffolds that we could find for each gene. So also highlighting the fairness of this approach is that we don't define a number of active scaffolds per gene. So we just mine the generated molecules, and actually for 4 out of the 5 genes, we didn't find any enriched scaffolds, so we couldn't perform any analysis on those genes. And for each of these genes, we found a variable number of scaffolds. And at the end, when we compared these scaffolds against active molecules, we saw that we could only find active scaffolds among active molecules containing these scaffolds for BRCA1 and for NF-kappa beta. So put it into numbers. For BRCA1, out of 8 proposed scaffolds was found in a potent agonist, which would be a hit rate of 12% compared to 7.6 in the training set. And this is, I think, remarkable because we're really proposing scaffolds from scratch without any information before. And for NF-kappa beta, we proposed 2 out of 25 scaffolds were found in agonists, with giving a hit rate of 8%, which is also considerably higher compared to the hit rate that we will find in the training set. And we wanted to still investigate how specific the generation of these active-like molecules was. So we generated a fixed amount of molecules conditioning the generator on different profiles from different genes. So here you see the genes that I included in the previous analysis, but I also now added the 10 most differentiable genes from this data set of 220 genes that we had available. Because now we don't need known actives for these genes, we just use them as a reference to see how often we generate agonist-like molecules. So, okay, that was kind of complicated, but at the end we have all these genes that we're checking for, and we generate molecules and quantify how many times conditioning in each of these genes do we generate molecules that are closely resembling these reference agonists that we have for BRCA1. And to define active-like molecules, we use 3 similarity thresholds, so on dissimilarity. And the same for NF-kappa beta. And what we find is that indeed the generation of these active-like molecules is very specific. So we have significantly increased counts when we condition specifically on BRCA1 compared to all the other genes. And of course, here we're checking against the BRCA agonist. And the same for NF-kappa beta, with only non-significant differences when we condition on another gene, so NF-kappa beta 1A. So at the end, these results really encourage the idea that the generation of these active-like molecules was attributed to the morphological conditioning, and it also exemplifies how our model can be used to generate bioactives for specific targets. And, okay, I think we're still good in time, right?
Yes, we are good. Yes.
Okay. And yes, and then the last part of the results are the interpolation results. So one of the advantage of the model, or like at least the type of representations that we're using, is that we are operating in 2 continuous spaces. We have a continuous feature space for the morphological profiles and a continuous feature space for the chemical descriptors. And therefore, that allows us to interpolate both spaces and see how the model behaves while interpolating the spaces. And therefore, we perform interpolation experiments around these agonists or active molecules proposed by the model. We did it for both for the BRCA agonist and for the NF-kappa beta agonist, but I'm just going to show the results for NF-kappa beta 1 for simplicity. So again, what we want to do is to do interpolation experiments to see how the model behaves around these predicted active molecules. And so how the interpolation experiments were done was we did interpolation in the chemical space, which is shown on the left, and also interpolation on the morphological space, which is shown on the right. And we start so for the chemical space, we start from a starting molecule, which is our reference agonist, do linear interpolation towards another molecule, and we use the condition network as a critic to tell us if the interpolated molecules, if the interpolated molecules match the NF-kappa beta overexpression profile or not. So this is an example of these interpolation curves. So we start with our known agonist, and we decided to interpolate towards another molecule, which according to the model is supposed to have a higher classification score towards the NF-kappa beta 1 profile. And what we see is that, so on the x-axis, we can see the cosine distance to the in the molecular embedding space to the starting molecule. And on the y-axis, this is our critic network, so the condition network reporting the matching probability towards our reference gene, which is NF-kappa beta 1. And what we see is that as we move progressively in the chemical space, so in the embedding space, we can see also discrete and reasonable changes in the chemical structures that are decoded from those locations in the space. And what we can also see, or is reassuring to see, is that these changes are not completely random, but there is a trend in when we move into the direction of this end molecule, the matching the matching of the condition network increases progressively. And the other experiments then interpolating the morphological space, what we do is we start from the DMSO morphological profile and do linear interpolation towards an end profile. And again, we use the condition network as a critic to tell us if the interpolated profiles match the a reference molecule or not. And again, the reference molecules against which we are computing the matching against is our reference proposed agonist towards NF-kappa beta, and the ending profile, so the direction towards which we move. We use 3 directions. We first move into random directions, into the top PCA directions, so the directions in the morphological space of maximum variation. And finally, we moved specifically towards the direction of NF-kappa beta, of the NF-kappa beta profile, which was the gene for which this molecule is supposed to be active on. And what we see is that as we start moving into that, we see first from the general trend, we see that there is a nice dose response as we move away from the inactive or the negative control phenotype. And what is very interesting is that indeed, when we move towards the direction of NF-kappa beta, we see a higher increase in the matching probability towards this agonist than if we move in the top 10 PCA components or in random directions. So this shows that the model is indeed learning some meaningful trends in the morphological space, and it doesn't rely only in general activity or general phenotypic strength as it is represented by the top PCA components. And we repeated these experiments for different molecules. So we also did the interpolation for this end molecule, finding similar results, and also try interpolations in the opposite direction. So not towards increased activity, which will be like molecular optimization example, but also towards decreased activity, just to see what happens. And we see that we get very similar results, although the classification curves for lower activity are more random, also because of the randomness of the end molecule that we picked. And when we do linear interpolation in the morphological space, in the opposite direction, we also see that when we use that when we move directly towards the NF-kappa beta 1 phenotype, we get a faster decrease in the classification towards this molecule. So at the end, from this interpolation experiments, what we can conclude is that they highlight the quality of the molecular descriptors and also the morphological descriptors. And it also shows that the morphology chemistry relationships learned by the model are kind of sensible and sound, since they show those response effects and they are not just isolated predictions. And this also demonstrates the potential of our framework for molecular optimization. All right. And
yes, can you go back to your previous slide? I was wondering if these results are more showing kind of more telling about the smoothness of the spaces that you have pre-trained than anything else, because like I can easily see it's working great, but at the same time, let's say we have like activity cliffs, like molecules that are very close, but then very different activity, then it will be very hard for the model to kind of it'd be easy for the model to fail to detect that, right? And propose like some of the activity cliff, like for example, let's say you want to optimize the activity, you say, okay, this one might be slightly better activity or have the same activity, but yet that's not the case because like it's just due to the smoothness of the space.
I'm not sure if I got the idea.
Yeah. So I guess the question is how much of this is due to the smoothness of the, maybe for example, in the case of the molecular space, like how much of this is due to the smoothness of the pre-trained autoencoder and rather due to things that's been learned by the overall network.
Yeah, that's a difficult question to to answer, but I think like the conclusions that we're drawing from here, okay, I said that the smoothness or this dose response behavior is reassuring and encouraging, but I think that the truly or less biased conclusion is about the steepness of the changes when you move selectively in different directions. So I think, yeah, the main take-home message is more that if we move specifically, like especially for the other case, just a second, for this case, that we move, that if we use this known agonist, if we move specifically into the direction of NF-kappa beta 1, we get a higher, more steep increase in the matching probability to the molecule than if we move in random directions or in the highest axis of variation. And yeah, but you're right. Like I cannot say from where this smoothness and dose response is coming from. It could be that is due to the variational autoencoder that learn the chemical representation space. Yeah.
Okay, thank you.
All right. So that brings me to the end. So as the main conclusions, we think that our work demonstrates that it is possible to influence the chemical space and propose tailored bioactives solely based in cell morphology and without prior information on biological targets. And what we also think is interesting from this approach is that based on it is based on a target-agnostic readout, so cell painting, and therefore it facilitates knowledge transfer between different biological pathways, and in principle it could be used to design bioactives for many targets under one unified framework, of course with the small letters only for those targets that show a distinctive phenotype in cell painting in the cell painting assay, which is of course not all the targets. And we think that this work provides a first step towards the systematic use of high content imaging for molecular design. And we still think that this is a proof of concept, and that by using larger data sets, we expect that we will be able to push our approach beyond this proof of concept and provide fast access to innovative chemistry influencing diverse biological pathways. And yeah, so that was all, and thank you for your attention, and I would like to take more questions or discussions, comments, anything.
Perfect. Thank you so much, Paula, for the great talk. If you have any question, please raise your hand or write in the chat. Yeah. Please go ahead, Daniel.
Hi, thanks for the talk. Very interesting. Kind of a basic question. I was just wondering why you chose the GAN generative framework over another, there's a lot of popular ones you highlighted, diffusion models early on. Just wondering how you arose at that.
Yeah, so we started with this project very long ago because it has been like a side project. So we started before diffusion models really came out and became popular. But another reason is that we already had a paper, actually in this, oh no, it's not there. We already had a publication in Nature Communications where we had proposed the same approach, but with gene overexpression, with gene expression profiles, so with transcriptomics. And therefore, we just basically adapted the code and architecture just to plug morphological features instead of gene expression features and repeat the same drill. So it was more a practical motivation.
Gotcha. Yeah, totally makes sense. Things go fast in that world.
Yes.
Any other questions? One thing that was bothering me when you showed the first set of results is the validity of the whole generation process, and I was wondering, like, what could you do, or could people do to kind of improve on this validity aspect? Is it mainly due to the low validity of the initial VAE, or is just because of the fact that this VAE is trained separately from the GAN? I'm just kind of trying to understand who is the fault. If is it the second model or the first one, or the fact that they are not just trained end to end?
So in this specific case, it was mainly due to validity of the autoencoder. So we tried to encode, decode molecules independently of the generator. And when we plugged in the generator, the validity reduced also compared to the autoencoder alone, but the autoencoder was already a bit suboptimal. So yeah, that was the main reason.
Okay, perfect. Thank you. There's a question in the chat from William. What is the typical throughput for doing cellular imaging? Means the beginning of the talk. Yeah.
What's the typical throughput for doing cellular imaging?
Yeah, I think it's high throughput, right?
Yeah. So it is like so at least if you have robotic systems performing the assays for you and you also have automated microscopes, you could easily test around 9,000 samples per week using only one microscope, one of these high throughput microscopes. But of course, these cell painting features are also noisy, and you're always recommended to do at least 3 repetitions per perturbation, preferably 4 to 5. So yeah, 9,000 samples per week, but you need to multiply by the number of repetitions, technical repetitions.
Okay, perfect. Jonathan?
Yeah, thank you. This was really interesting, and I mean that in the American English usage, which is a positive versus the British English. So what would you compare this to? Like, help me contextualize this. Like, these methods are really cool. It's very interesting. You know, the generative methods seem to perform, pretty well. But, it seems like you're generating a bunch of molecules. You're looking at the scaffolds and you're saying, how similar are these scaffolds to things that I know work? And how could this be used, by Bayer or somebody, or what would this be pit against, so that you could say that this is much better than, something else?
Yeah, like if I will need to prove the managers at Bayer that this works, I will pick one of our projects, internal projects, where the chemists are trying to design and optimize the hits and test the target that they are searching for in the cell painting assay, ask this model to generate molecules and find which scaffolds are enriched for that specific morphological profile, and then ask the chemist to also synthesize the proposed molecules by the GAN and compare if they are active or not. So, and I think, so that will be how to prove that this actually generates decent molecules for the specific target. But then the next step, it would be to identify for which genes, or at least for which targets in the morphological profiling space, we are sure that we get distinctive morphological profiles such that we can trust the generated molecules, and then really start, let's say, de novo testing for different projects. But first, it will be to identify for which parts of the morphological space do we get decent and reasonable predictions. And yeah, and that could start new projects. If you say, out of nowhere, I think that these molecules are going to hit this target with very high confidence. And yeah, that could create new projects. Does this answer the question? Or?
Yeah, I think so. I mean, let's say that I were to give you a new target and say, you know, give me the 100 best compounds that you believe in, and I'm going to pit you up against a bunch of, you know, very skeptical medicinal chemists that are going to do the same. And, you know, let's say we don't actually have starting material, so we can't go and say, well, these scaffolds are similar to what we know. You know, what would be like the hit rate, expected hit rate for something like this? Or is that just too, you know, this is way too, you know, proof of concept right now to even answer something like that?
Yeah, this is very proof of concept, but I have to say, so for some of the genes, we selected very few molecules and we synthesized those molecules and tested them against the targets. And what we found is that, I don't have the numbers here, but it was like really, we got an unexpectedly high activity rate of these synthesized molecules against the targets. But the puzzling thing was that they were not agonists. Most of them had an antagonist effect. We were like, oh, okay, interesting. But at some point, also some chemists said, like, actually the line between agonism and antagonism is very thin, because sometimes you modify something in a molecule and then it becomes an antagonist instead of an agonist. Especially agonists are very tricky.
Yeah, it seems the one constant of all these models is that they'll surprise you. Anyway, thank you very much. Yes. Very interesting time.
Thanks.
I think there's a question by David in the chat. He's currently involved in setting up a cell-painting assay for a compound library. And he's wondering if you have noticed any compound class being underrepresented in the current available data set.
No, I didn't really explore so much the chemical space of the current data set, but I mean, so what are you trying to do? Are you trying to create a diverse library for cell painting? Because I could refer you to new resources, like I don't know if you're aware of the JumpCP consortium, which was a large consortium where we screened 130,000 genetic and chemical perturbations in a collaborative effort between several nonprofit and profit organizations. And if you start using that data, there, like, only chemical perturbations is I think something like 120,000. So there you could really use this data to define a phenotypically diverse, like phenotypically and chemical diverse set. But I didn't explore that, the diversity.